Thank you very much. This morning I'm going to talk about a subject that's rather different than what I've spent my career working on. Instead, I focus on a very different, even more challenging problem than vesicle traffic, and that is the challenge of neurodegenerative disease. Parkinson's disease is the most rapidly rising neurodegenerative disease in the world now, exceeding the pace that we see with Alzheimer's disease, although, of course, there are still more Alzheimer's patients. It's rising extraordinarily rapidly around the world. It observes no boundaries. It is like a pandemic. Its rise in recent years may be attributed to environmental toxins. Only a fraction of the disease can be explained by mutations. We now know of over 20 different genes, mutant alleles of which predispose to familial forms of Parkinson's disease. It's like a puzzle, only much more complicated than that. We know of the genes and the proteins that they encode that afflict patients who have a genetic form of the disease, and some of these can be explained by the pieces of the puzzle that you see before you. In the upper right-hand corner, I'll tell you about one such example, a gene that encodes a brain-specific protein called alpha-synuclein. There are others, but the picture is going to be much more complex because the genes, as we know them, have no obvious connection to one another. They affect other pathways. Ultimately, these pathways are known to impinge on a region of the brain, the midbrain, the substantia nigra, where a cluster of dopaminergic neurons are found that are responsible for controlling executive function, movement, and which are, as you'll see, particularly vulnerable to these mutations, but likely also to environmental toxins. It's going to take an enormous effort to uncover the reasons for this disease. This disease was first described by a British physician 200 years ago, and in that time, nothing has emerged that is effective in treating the disease. The treatments that we have are only palliative. They only block some of the symptoms. But the arc of the disease is inevitable and inexorable and ultimately leads to the death of the patient. Now, many of you will know about the brain at some level. Here's a cartoon of a nerve cell that you may have a mental image of in your mind. There are, see, this mouse doesn't really work, so I'll just have you imagine the middle of this cell is the cell body where the nucleus is. Many of the organelles that perform housekeeping functions are located there. To the left are dendrites that receive information from other cells, and then to the right, and to the left, there's a long axon, an axon that can extend as long as a meter up and down the spine, terminating in nerve terminals that are the means of communication between cells through the secretion of chemical neurotransmitters. Now, this is a typical image of a nerve cell, but let me show you why the dopaminergic neuron is particularly vulnerable to both environmental insults and to mutations. Here's an image of the dopaminergic neuron. In the substantia nigra, there represent perhaps tens of thousands of such cells. They die at some rate in all of us as we age, but in Parkinson patients, they die somewhat more rapidly and progressively. It's estimated that by the time a patient presents with the movement disorder symptoms, over half of these dopaminergic neurons have died. And then the inevitable outcome is the final death of these neurons, as you'll see in an example in a moment. Now, the dopaminergic neuron has also a cell body, as you see in the lower left, and some dendrites and a single axon. But look at the complexity of the web, the forest of nerve terminals. It's estimated that 1 million or more nerve terminals must be guided up to the upper regions of the brain to connect to an area called the striatum that controls executive function and movement. These cells are always on. They're always being stimulated. They're always at peak performance. Their mitochondria are functioning to produce as much ATP as they can possibly sustain, and anything that does damage to the mitochondria or to the function of the cell makes it much more vulnerable. That may be why the mutations, one of which I'll describe, seems to be somewhat selective in the demise of dopaminergic neurons and possibly better tolerated by other cells. Now, You might ask, why would I be interested in the dopaminergic neuron or in Parkinson's disease? Well, let me tell you why. We know that one of the proteins that's implicated in this is a protein called alpha-synuclein. Here you see sections of the midbrain of two patients, one of whom died for natural causes. This person is labeled C on the right of the left-hand panel. In the midbrain section, in unstained samples, you can see a band of cells, the darkly stained band that represents the substantia nigra. That's because dopaminergic neurons, in producing dopamine, produce a byproduct called melanin, which is the normal pigment in our skin that is responsible for color absorption. This pigment is just a natural outcome in humans, rather specific for humans, that allows one to see the vitality of this tissue, of course, only in a section from a deceased patient. On the left is an image of a cross-section of the midbrain of a patient who died of Parkinson's disease. Very little dye and very little melanin remains, and this patient has almost certainly died as a result of the loss, almost complete loss of the dopaminergic neuron. On the right, about 100 years ago, a German clinician by the name of Frederick Louis was able to visualize this substantia nigra region in a patient who died and found inclusions of structures, these darkly stained structures, that we now know appear to be initiated by a misfolded form of alpha-synuclein that has the tendency to collect around it other proteins and fibers and membranes to create this enormous structure that is now called the Lewy body. Unfortunately, this Lewy body can only be visualized in sections of deceased patients, so we aren't able to follow it during the progression of Parkinson's disease. Nonetheless, in a number of sections that have been conducted, it's clear that there is some progression of this structure in the brain. It may be that the structure itself is the pathologic basis of the demise of dopaminergic neurons, or this may simply be a byproduct of a process that leads to the death of these cells. Another German clinician in the 20th century discovered in sampling many more brains, sections of brains of patients who died, a seeming progression of these Lewy bodies. You see a patient who died possibly because of Parkinson's at an early stage, where the Lewy bodies appear at the brain stem and may even project into a region on the left corresponding to the olfactory bulb. And it is a possible correlation that the appearance of this structure is in a tissue that may affect patients very early on before the other symptoms of Parkinson's disease emerge. People who eventually progress to Parkinson's disease recognize that many years earlier, possibly decades earlier, they experienced a sudden and quite substantial loss in their ability to smell scents. As the disease progresses, Lewy bodies can be found elsewhere in the brain, including in the midbrain, possibly in the higher cortical regions that produce other symptoms, sleep, and the most profound defect of motor disturbance. And finally, in the end stage, these structures can be seen in the cortex, where they very often, a majority of patients, often experience dementia as a result of the progression of the disease. Patients can survive for decades. Michael J. Fox, the famous actor in the US, is now nearly 40 years into his term as Parkinson's. He is severely afflicted, but he survived a great length of time. Others succumb to the disease much more quickly. Now, as to my own particular interest, it was partly professional, but largely personal. And let me share with you the story of my wife, Nancy. We were married in 1973 in a park near Stanford. We had a wonderful life together. We moved together to Berkeley, where our children were born and raised. Everything was great. Things were looking swimmingly well, until at the tender age of 48, she presented with some strange symptoms that at first weren't interpretable. But ultimately, it became clear that she was suffering from the early stages of Parkinson's disease. We were able to suppress this disease by a dopamine replacement medication that most patients take early in the disease, but the treatment wears off very quickly, and within 5 years, patients develop other symptoms, other pains, rise and fall in mobility, a loss of balance, very often sleep disturbances, the awkward dyskinetic movements that you see in these patients. Nonetheless, life went on. Our children went off and graduated and went on to their own lives and careers. There were some happy moments. Here, for instance, is a precious picture that I have in my home of Nancy and my dad at the Nobel banquet in October of 2013. She was at this point already suffering from dementia, quite serious dementia. I was grateful to have my family there to care for her when she couldn't care for herself. But within the next few years, it got worse and worse. She was unresponsive, sometimes catatonic. She could not feed or bathe or clothe herself. It was difficult to give her medications in the morning. She would sleep all day long if I didn't wake her up. I had access to the best neurologists and experts in her particular form of Parkinson's around the world in my travels. I knew that there was nothing that I could do, that there had been no significant progress in the treatment, but I always asked, how could I prepare myself? What could I do next to try to comfort her? I was always given an answer, and they were always wrong. That's the frustration of support groups and family members who must care for a patient with such a disease. Parkinson's is like a spectrum disorder. It presents at different symptoms, it progresses at different rates, and ultimately patients die. She died in the middle of the night, now 8 years ago, likely of heart failure. I was never warned that that was a possible outcome, but patients often have a pathologically low blood pressure, and they pass out and they fall, they hit themselves, and in her case, it was likely that her heart muscle lacked oxygen perfusion and thus gave way in the middle of the night. Well, out of this tragedy came something quite unexpected. Just shortly after she died, I was approached by the chief financial officer of the Sergey Brin Family Foundation. Mr. Brin is the co-founder of Google, enormously wealthy, still healthy himself, but he shares a dominant mutation, a dominant allele of one of the 20 genes, a gene called LRRK2, LRRK2. He shares that mutation with his mother, who has advanced Parkinson's and is quite ill. Mr. Brin is still healthy, but he is at considerable risk. As a result of that knowledge, he's invested hundreds of millions of dollars in the Michael J. Fox Foundation, which is a wonderful charitable organization in the US that supports patient groups and clinical research. But they have a very small budget for basic science. And Mr. Brin decided on his own that in the 200 years since the disease developed, we still didn't understand why those dopaminergic neurons are so vulnerable. He felt that what we needed was an investment, a significant investment, in the basic science of Parkinson's disease, that in order to discover the molecules and the cells where the disease originates and how it progresses, that we would then find new clues and new medications that might treat individual forms of the disease. So out of this grew a commitment that I made to really change my life, to help found an organization called Aligning Science Across Parkinson's Disease. This is an organization that I helped found that has settled on funding groups of scientists, teams that have already established collaborations, either on Parkinson's or on a related theme, teams all over the world that agree not only to cooperate within the group, but among the teams, and they would be funded quite significantly to push the goals forward in a collegial and collaborative manner. We focused on 4 themes that we felt were essential in understanding and achieving the desired targets that we needed to know. We focused on the genes and their protein products, the associated biology, only one of which I've given you. There are many others and many obvious things that could be done to understand how these seemingly unrelated genes interrelate and lead to the death of a dopaminergic neuron. There are aspects of the immune system, both of the brain and of the body, that seem to initiate or possibly exacerbate the lesions that are found in the brain through inflammatory processes that are poorly understood. The cells itself are enormously complicated, as I've already given you a snapshot of, and the circuitry, the innervation of those million nerve terminals is far from understood. There is more basic understanding that we need. And finally, there's a curious prodromal phase before the onset of the movement disorder, that often accompanies progression. I told you about one, a poorly developed sense of smell. Another very curious phenomenon is called REM sleep disorder. This is where patients lose their ability to suppress movement during nightmares while they're sleeping. They often are so violent that they throw themselves out of bed or they start fighting with their partner. It's really a very frightening ailment. And yet, even worse is that within 5 years, 80% of these patients progress to Parkinson's disease. Well, let me give you a snapshot of where we are. We are now at the midpoint, 5 years into a 10-year program. With Mr. Brin's generous donation of funds, we've already invested over $500 million. At its peak, we had 35 teams, 163 investigators, one of whom is sitting on stage, Uli Hartl. We have a wonderful balance of teams. Each team has to have an early career investigator. We are represented in 14 countries around the world, 80 different institutions, and quite a number of women taking leading roles. Almost all of our interactive work is done online, but every year we have a wonderful meeting. On the right is an image of our first meeting in the Bahamas in 2022. I'm standing in the front row next to a key person in the team, and that's a woman named Kemeny Riley, who is the executive director of our program, and flanked on our left and right are leaders of the Michael J. Fox Foundation, with whom we work hand in hand. At these meetings, each of the PIs come together to meet each other, to form new collaborations, and to exchange ideas, reagents, results. All of this collaborative effort, I'm confident, will in the end of our program lead to new targets that will be taken up by the pharmaceutical industry, with the hope that we'll have much more effective treatments in the few years ahead. Thank you.